Hello and welcome to another episode of Momento Malum today. Interspecific hybridization is a commonly used method to increase the size of a crop species gene pool. Breeders have used interspecific hybridization to obtain genes to improve the crop's tolerance to various environmental and pathogenic stresses, as well as enhance economically important traits such as quality and yield. Successful outcomes of interspecific hybridization can be seen in blueberry breeding. Interspecific hybridization is the crossbreeding of different species to improve the gene pool, such as crossbreeding a blueberry with its wild relatives. Indeed, the cultivated highbush blueberry has been bred with other species such as the cold-hardy lowbush blueberry, the wild highbush blueberry, and the southern rabbit-eye blueberry. The more northern breeds have wild highbush blueberry and lowbush blueberry pedigree mixed in, whilst the rabbit-eye blueberry is used in southern cultivars. Blueberry breeding and domestication only began in the last century. Highbush blueberry domestication and breeding began in the 20th century, specifically in the United States. Interspecific hybridization of blueberry species has played a major role in the development of highbush blueberry cultivars. The Southern Highbush Blueberry Group, also known as the SHB Group, is a cultivar group of blueberries better adapted to warm climates than the original Northern Highbush Blueberry or NHB Group. The SHB Group was derived from crossbreeding the NHB group with wild blueberries native to Florida, such as the scrub blueberry and the rabbit-eye blueberry. As part of the breeding experiments to improve heat tolerance in southern lines, every southern cultivar has at least one Florida wild species in its pedigree. The union between two Japanese universities and USDA would analyze many southern blueberry cultivars to get a full breeding history of the southern lines using SNP genetic marker technology to analyze the genetics and hear the results. But first, ad time. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below. The genetic analysis revealed that the cultivated rabbit-eye blueberries form their own distinct genetic cluster. The NHB cultivars also cluster together, with some exceptions. Some of the NHB lines were genetically far from the NHB cluster, and some of the SHB lines were found to be in the NHB cluster. Specifically, the NHB cultivars Blue Crop and Bounty were found far from the NHB cluster. The half-high cultivars, which are hybrids between the low bush and the high bush blueberry, and exhibit cold hardness, clustered with the NHB cultivars, with the exception of Top Hat, that was far from the NHB cultivars. Another group of cultivars called the CH cultivars, which were bred with the Southern Lines group, grouped with the SHB cultivars. There's also a clear separation between the rabbit-eye and highbush blueberry cultivar groups. A deep relationship between the SHB and NHB group was also found, and is likely explained by the continued mixing between the SHB and NHB cultivars. It was also found that there are some southern blueberry cultivars with minor traces of half-high blueberry pedigree. Overall, the researchers concluded that the northern lines are more homogeneous, genetically speaking, than the southern species, due to more mingling between different lines of blueberry, both southern and northern, as well as the continued crossbreeding between these lines and wild Florida blueberries. Despite the extensive breeding and admixed nature of the SHB population, there appears to be no genetic segments within its population that are common to all SHB cultivars. And that about covers everything. Thank you for watching.